Good morning, dear students. Today we are going to learn about the uniparental disomy. Now, what exactly is uniparental disomy? It is, in short form, you can call it as UPD, which occurs when person receive two copies of chromosomes or part of chromosomes from one parent and do no copies or have no copies from other parent. Okay, so the person is having chromosome set from only one parents. Suppose he receives from father both the sets, he will not receive from mother. So UPD can occur as a random event during the formation of egg or sperm cells or may happen in early fetal development. In most cases, UPD does not affect development of a person as most of the genes are not imprinted. So it doesn't matter if a person inherit both copies from one parent instead of one copy from each parent. Okay. So in early development, basically these things happens because uh, though in early development the cells are not expressing the genes. Okay, so in that matter it will not get imprinted in the developmental process. Instead, later on, when it becomes imprinted in the later stage, it shows the effect. Okay, but in some cases, if a gene is inherited from person's mother or father, it causes genetic imprinting. Genetic imprinting takes place when one of the two copies of genes is active. Okay, which copy is active depends on the parent of origin. Some genes are normally active only when they are inherited from the person's father. Others are active only when inherited from a person's mother. And this phenomena is called genetic imprinting. A person with uniparental disomy may lack any active copies of essential genes that undergo genetic imprinting. For example, this Angelman syndrome and Prader-Willi syndrome. Okay, in short, it is called as Angelman syndrome (AS) and Prader-Willi syndrome as PWS. These are the two examples of disorders that can be caused by UPD. Angelman and Prader-Willi syndromes are neurobehavioral disorders most commonly due to large deletion of chromosome 15 Q11 to Q13. Okay, you can read it as 15, that means chromosome number 15. In that, Q arm and bands 11 to band 13. The deletion arise differently during the maternal or paternal gametogenesis. So, in short, when the person receives two copies of chromosomes or part of chromosome from one parent, then only uniparental disomy occurs. So, let's first study the Angelman syndrome. Okay, Angelman syndrome is a genetic disorder that primarily affects the nervous system that causes developmental delay and neurological problems. Okay, Angelman syndrome, which is a genetic disorder and causes the nervous system and developmental delay in neurological problems. Okay. Angelman syndrome occurs when a person inherits both copies of section of chromosome 15 from the father, or when chromosome 15 is inherited normally, one chromosome from each parent, and the gene in the mother's chromosome is deleted. Only father's section will be present. And this deletion of section of maternally inherited chromosomes is the most common cause of Angelman syndrome. Okay, and now you can easily understand that the male or father donates just one sperm. Okay, the mother is giving the providing the entire what shelter to developing embryo. Okay, so the maternal effect is always more. So there are some kind of maternal effects that can be seen during the development, and if suppose the maternal genes are not present or chromosomes are not present in any person, they are going to affect the development process. Angelman syndrome is named after the physician Harry Angelman, who first described it in 1965. It affects an estimated 1 in 12,000 to 20,000 people. Okay, the person suffering from uh, this Angelman syndrome are hyperactive and have short attention span. Okay, they cannot concentrate, they cannot give attention to anything. They are very hyperactive, एकदम शीघ्र कॉपी म्हणतो ना आपण त्यांना तसं. They have unusual facial appearance, short in height, microcephaly, then protruding tongue, and they have sleep disorders and show jerky movements. These are some of the symptoms of this Angelman syndrome. Okay, so next is the this Prader-Willi syndrome. Prader-Willi syndrome, that is PWS, occurs when person inherits both copies of section of chromosomes 15. This deletion section of chromosome 15. Okay. Now this deletion largely takes place in the father, and genetic imprinting plays a role. In child with PWS, that is Prader-Willi syndrome, the activities of some genes in the region 15q1 to q13 on the maternal chromosomes shows problem. 15 normally are suppressed as a result of genomic imprinting, and this syndrome occurs in about 1 in 25,000 births. What are the symptoms of this Prader-Willi syndrome, which includes small and weak at birth, weak cry, mental retardation, poor feeding caused by difficulty in swallowing and sucking the sucking reflexes. They have short height, small hands and feet. Okay, so these are some of the symptoms of these two syndromes. That is the Angelman syndrome, and this is the Prader-Willi syndrome. Okay, so how to diagnose these syndromes? Diagnosis is a very important thing for uh, important thing in case of this genetic disorders. Okay, how you can come to know that whether these particular symptoms are due to genetic disorders, and uh, if there is understanding about this thing before birth of the individual, you can either treat, or you have to decide whether you should go for abortion or any other thing. Okay, so let's study about the how diagnosis of this uh, diseases or disorders. Genetic tests are done by analyzing small samples of blood or body tissues. They determine whether you, your parents, or your baby carry genes for certain inherited disorders. Genetic tests don't yield easy to understand results. They can reveal the presence, absence, or malformation of genes or chromosomes. Okay, so if the parents, they will take this genetic test, they will understand about any inherited disorders in their baby. Okay, and then depending on the results of all these malformation of genes or chromosomes, they can they can decide whether they should go for pregnancy or not. Okay, deciphering what these complex tests mean is where a genetic counselor comes in. Okay, now amniocentesis for parental diagnosis, that is the amniocentesis is also called as amniotic fluid test or AFT. Now, what is this amniotic fluid amniocentesis that we will see now? It is a medical procedure used in parental diagnosis of chromosomal abnormalities and fetal infections, in which small amount of amniotic fluid. Now, where this amniotic fluid is located, you can see it in this particular picture. Okay, this is the amniotic fluid which acts as a shock absorber, and it gives the developing baby protection from any external mechanical shocks. Okay, and this amniotic fluid can be removed from the amniotic cavity. So it is a medical procedure used in parental diagnosis of chromosomal abnormalities and fetal infections, in which a small amount of amniotic fluid which contains fetal tissue is sampled from the amniotic sac surrounding a developing fetus, and then the fetal DNA is examined for genetic abnormalities. Okay, this is very old technique, and now there are different techniques like scanning and ultrasound techniques are there, through which they can study about any abnormality in the developing fetus. The most common reason to have an amnion is the determine whether a baby has a certain genetic disorder or chromosomal abnormality such as Down syndrome. Amniocentesis can diagnose these problems in the womb. Okay, it can they can diagnose the problems in the womb, and amniocentesis is preferred when the woman is between 11 and 16 weeks gestation. 14 ते 16 आठवडेच्या जेस्टेशन पिरियड नंतर काय घेऊ शकतो आपण ॲम्निओसिंथेसिसची टेस्ट घेऊ शकतो. वुमन हू चूज टू हॅव this test are primarily those at increased risk of genetic and chromosomal problems, in part because the test is invasive and carries a small risk of miscarriage. Now, just see the procedure, what they are doing in this particular procedure. They are injecting a syringe with a needle and taking out the this amniotic fluid. But after seeing this procedure, you can easily make out it may create risk with the developing fetus. ती सिरिंज आहे ती आतमध्ये काय करू शकते, प्रॉब्लेम क्रिएट करू शकते, किंवा त्याची निडल आहे ती डेव्हलपिंग फिटसला पण लागू शकते. This process can be used for parental sex discriminant, and hence this procedure has legally restriction in some countries. Legal restrictions are there because they can easily make out whether the developing baby is male or female. Okay. आणि पूर्वीच्या वेळेला बर्थच्या आधीच डॉक्टरना जेव्हा amniotic सिंथेसिसची टेस्ट केली जायची त्यावेळी ते कळायचं आणि त्यामुळं पूर्वी खूप लहान मुलांमध्ये हे डिस्क्रिमिनेशन केलं जायचं मेल आणि फिमेल फिटसमध्ये आणि फिमेल फिटस असेल तर त्याचं ॲबॉर्शन केलं जायचं आणि त्यामुळं बऱ्याच कंट्रीजमध्ये हा जो ही जी प्रोसिजर होती ती काय केली गेली बॅन केली गेली or रिस्ट्रिक्ट केली गेली. The doctor insert a hollow needle in the woman's abdomen to remove a small amount of amniotic fluid from around the developing fetus, and this fluid can be tested to check for genetic problems and to determine the sex of the child. When there is a risk of premature birth, amniocentesis may be done to see how far the baby's lungs have matured. ओके. काही वेळा प्रीमॅच्युअर बर्थ होतात म्हणजे युजली 9 महिने लागतात बर्थ व्हायला खरं त्याच्या आधीच 6 महिने 7 महिन्यालाच काय व्हायला लागतं की प्रेग्नन्सी होते आणि त्यामध्ये ह्या टेस्टचा उपयोग असा होतो की याच्यामुळं आपल्याला समजू शकतं की बेबीच्या लंग्ज आहेत ते डेव्हलप झाले आहेत काय की आउटसाईडला घेतल्यानंतर बाहेर काढल्यानंतर ते सर्वाईव्ह होऊ शकतं काय याची आपल्याला कल्पना येऊ शकते. ओके. द नेक्स्ट टेस्ट विच इज अगेन व्हेरी इम्पॉर्टंट दॅट इज द कोरियोनिक विलस sampling. Okay. Chorion is another membrane, extraembryonic membrane, and this chorionic villus sampling, that is CVS, is another method. In this procedure, a catheter is passed via the vagina and through the cervix into the uterus to the developing placenta under ultrasound guidance, or the alternative method is transabdominal. Okay. The introduction of the catheter allows sampling of cells from the placental chorionic villi, which can then be analyzed by a variety of techniques. The most common test employed on cells obtained by CVS is chromosome analysis to determine the karyotype of the future fetus. The cells can also be grown in of culture for biochemical or molecular biology analysis. CVS can be safely performed between 9.5 and 12.5 weeks of gestation. Okay, so two methods we have seen at present. The third method, which is again very important method to understand about the genetic disorders, if any, and that is about the banding of chromosomes. Now, in case of CVS, has a disadvantage also of being an invasive procedure, and it has a small but significant rate of morbidity for the fetus, and this loss rate is about 0.5 to 1% higher than for women undergoing amniocentesis. Okay. Rarely, CVS can be associated with limb defects in the fetus. The possibility of maternal RH sensitization is present, and there is also possibility that maternal blood cells in the developing placenta will be sampled instead of fetal cells. ओके. म्हणजे कदाचित प्लॅसेंटामध्ये मॅटर्नल ब्लडचं सॅम्पल येऊ शकेल इन्स्टेड ऑफ जे बेबी असेल डेव्हलपिंग फिटस असेल त्याच्या ब्लडऐवजी, विच कॅन बी व्हेरीफाईड विथ द क्रोमोझोम ॲनालिसिस. म्हणजे त्यामुळं काय होऊ शकेल आपला जो ॲनालिसिस आहे तो चुकण्याची शक्यता पण त्याच्यामध्ये नाकारता येईल. Then another technique which is again very important, that is the banding of chromosomes. Okay, a number of technique demonstrated the banded appearance of chromosomes. You must have seen the picture of chromosome, and in that you can see that there are bands on the chromosomes. The general banding technique shows a full range of bands throughout the chromosome complement. Okay, but special banding techniques, procedures, or it produces a restricted staining of one or other subsets of bands. म्हणजे ज्याच्यामध्ये भरपूर बँड्स दिसत असतात क्रोमोझोममध्ये. खरं ज्या टेक्निक आहेत त्याबरोबर काय करतात, पर्टिक्युलर बँड्सना काय करतात, स्टेन करतात. म्हणजे वरच्या ह्याच्यात तुम्हाला दिसते बघा, स्टँडर्ड टेक्निक, स्टेनिंग टेक्निक आणि जी बाइंडिंग टेक्निक. ओके, so binding techniques reveal many morphological features and finer details of the chromosomes by which chromosomes can be identified. Okay. At Paris Conference in 1971, a system of nomenclature was established that can be used to uh, designate the position of any band or interband in given chromosome. Special stains that bind preferentially a certain segment of DNA because of the presence of higher or lower concentration of certain constituents. Procedures, certainly visible patterns of bands. Bands are the crosswise strains or striations on the chromosomes. ओके, म्हणजे हे जे स्ट्रायशन्स दिसतात तुम्हाला, पट्टे दिसतात, तर ते बरोबर आयडेंटिफाय करणारी स्टेनिंग टेक्निक्स डेव्हलप केली गेली. क्रोमोझोम बाइंडिंग वॉज डिस्कवर्ड बाय टी कॅस्परसन. ही स्टेन्ड chromosomes with a DNA binding fluorescent compound, that is the quinacrine mustard. ठीक आहे, फ्लोरोसंट आहेत आणि because of that stain it gets stained properly, and that's why it is called as Q binding. Okay, different regions along the length of each chromosome shows a varied intensity of fluorescence so that the chromosome can be individually identified. Okay, so let's first see the Q binding technique. Q binding technique, it involves the use of fluorescent stains such as the this quinacrine mustard. When examined by fluorescence microscopy, each pair stains in specific manner of the bright and dim bands called as the Q bands. Q binding pattern is same in mitosis and meiosis. Now, another technique, that is the G binding technique, which is mostly used for this staining of uh, chromosomes. And the name is only because of the Giemsa stain we use. The Giemsa stain procedures or produces the identical binding patterns when used on variously pre-treated chromosomes preparations. G binding pattern differs from Q binding in one respect. G binding shows single additional band near the centromere or chromosome number 1 and 16, which is not shown by Q binding. This is specialty of G binding techniques. Now this G binding pattern is same in mitosis and meiosis, and it is very commonly used techniques. Here the chromosomes are treated with trypsin prior to staining with Giemsa stain. Now what is this trypsin? Trypsin basically is it's a kind of chemical or enzyme which degenerate the bonds between the cells. ओके, म्हणजे त्या सेल्समध्ये जे एकमेकांना सेल्स चिकटलेले असतात तर ते सेपरेट करण्यासाठी ट्रिप्सिनचा वापर केला जातो आणि त्यामुळे सेल्स सेपरेट होतात. क्रोमोझोममध्ये असणारे जे क्रोमोझोम एकत्र असतील तर ते सेपरेट करण्याचं काम ट्रिप्सिन करतं आणि त्यामुळं जेव्हा तुम्ही कॅरिओटाईपचा एखादा फोटोग्राफ बघितला तर तुम्हाला काय दिसेल, तर ते सेपरेट झालेले दिसतील. म्हणजे त्यामध्ये तुम्हाला जर स्टेन केलेले सेल असतील तर तुम्हाला कॅरिओटाईप मग त्याच्या साईजप्रमाणे तुम्हाला काय करायला लागतील, सेपरेट करायला लागतील. ज्याची लेंथ जास्त आहे ते एक वरच्या नंबरला येतील आणि ज्यांची लेंथ शॉर्ट होत गेले, जे शॉर्टेस्ट आहेत ते सगळ्यात शेवटच्या नंबरला आहेत. तर अशा प्रकारे तुम्हाला क्रोमोझोम नंबरिंग करता येऊ शकतं आणि त्यासाठी तुम्हाला त्या ह्याच्यातनं जे क्रोमोझोम सेपरेट झालेले आहेत ते तुम्हाला कट करून एका ह्याच्यामध्ये पेस्ट करावं लागतील अँड देन यू विल अंडरस्टँड अबाउट द स्ट्रक्चर ऑफ क्रोमोझोम्स. द क्रोमोझोम stains in a pattern of dark and light bands called G bands, with dark bands corresponding to the fluorescent bright Q bands. Okay, protein interferes in staining and hence trypsin is used to denature the proteins. Okay, then next technique is the R binding technique. This technique, when तर ह्या डायग्रॅममध्ये तुम्ही बघा, ॲक्च्युली तुम्हाला सांगायचं म्हणजे इथं तुम्हाला दाखवलं की सेंट्रोमियर आहे, राईट साईडला पी आर्म आहे आणि दुसरा या क्यू आर्म आहे, बरोबर आहे. देन हा जो हे काय दिलेलं आहे त्यांनी रिजन दिला आहे 1 आणि 2. तर सेंट्रोमियरपासून तो रिजन मोजला जातो, त्याच्यात सगळ्यात जवळचा जो भाग आहे तो 1 होतो आणि त्याचा लांबचा आहे तो 2 होतो. इथं क्यू आर्ममध्ये पण 1 आणि 2 असं दिलेलं आहे. Okay, now about the banding techniques, you can see it in this picture also. You can see easily. This is P arm, this is Q arm, then this is region 1 and region 2. Okay, and then these bands are named 1, 2, 3, and 1, 2, 3 like that. They can be named. Okay, here also you can see the one. So you can denote it as P11, or in that also some subsections are there which I have not shown here. So this is the landmark band, जो तुम्हाला डल दिसतो तो landmark band आहे, and this is the dark band. Okay, this is telomere, this is centromere. Again this landmark band, telomere in this particular picture. जे डार्क बँड्स आहेत ते आणि लँडमार्क बँड्स असे दोन वेगवेगळे बँड तुम्हाला बघायला मिळतील. Now next is the R banding and the C banding, and then we are going to finish this. So 27 minutes, we'll stop and later on we can conduct. Okay, but just finish the R band and C banding techniques. R banding, that is the reverse banding when pre-treatment with heat is given prior to Giemsa staining, that is first hitting the chromosomes, then treating with trypsin, and then giving the Giemsa stain. The resulting dark and light bands are the reverse of those produced by the Q and G bending. And the last bending technique, that is the C bending technique. The C bending can be brought about by alkali extractions of DNA from the chromosomes and highly repeated DNA sequences resist such extraction, and C bending can be brought about by staining with 80 specific DNA. Okay, and these techniques stain a small region adjacent to the chromosomes of all chromosomes, and also the distal portions of Y chromosomes. Using Paris nomenclature 1971, identification of band interband position is done, and the parameters used are: one, chromosome number, okay, whether it is 10, 15, 9, okay. Second, the arm symbol, that is P arm or Q arm. Region and bands number, that is the 7p23, pronounced 7p23, indicates the band 3 in the region 2 of the short arm of chromosome 7. P is the short arm. Okay, so like that you can easily go for the numbering of chromosomes or banding of chromosome. we'll conduct another lecture in some time, and in that we can finish about the this fluorescent test, that is the fluorescence in situ hybridization, and the remaining, that is the protein transcation test. Okay, and the last part remaining is the genetic counseling. So today itself we are going to finish this unit, and then we are going to start another unit. Thank you.